SpatialData, a fair framework for multimodal spatial omics, presented by Luca Marconato. Let's jump directly into the problem. Supposed to have a Visium spatial transcriptomics dataset. This is a type of technology which gives you gene expression recorded in space in circular capture locations. The data stored on disk is in the native format, and from this it's very easy to proceed with tasks such as visualization. It is also very straightforward to proceed in the analysis of the gene expression with external tools, for instance, cell to location, and these tools will then save the results back to disk. So imagine that you run a tool on gene expression and a tool that is working on the image part. These two tools will save the data on disk, but at this point, it's very common that the data that is saved is represented in a different way. So the new object that you have is not anymore the native object, and if you want to proceed with tasks such as visualization, it's not easy anymore to run the visualizer tool on the now processed data. And if you continue with downstream analysis, for instance, let's imagine that you want to run an external tool such as Giotto, written in the programming language R, on this type of data. Not only it is not easy to run this tool on this new combined data types, but also the result from Giotto will be R-specific, increasing the complexity of data representation. And the example that I just illustrated is for Visium 10x data. But right now, there are dozens of different available methods from various spatial omics companies. And what we see is that very often, each company comes with its own format, making it very complex to operate on this heterogeneous of datasets. There is also another level of complexity that we see nowadays, multi-omics spatial datasets, in which not just one technology is being analyzed, but the biologist employs an array of different techniques for the same or for overlapping tissue regions. And here is an example in which 10x Genomics presented a dataset, breast cancer dataset, combining one Visium field of view and two Xenium field of view. And from these different type of technologies, one can derive different type of biological signals and annotations. To solve this representation problem, we introduced a library called SpatialData. This is a framework that allows you to combine a variety of spatial elements, such as images, segmentation masks, also called labels, single molecule points, polygons, or other shapes, together with tables annotating these various geometries, and you can combine any combination of these spatial elements. The data that the user often finds comes from standard machines, for instance, from the 10x experiments or Xenium experiments. So we also developed a satellite library that allows the user to read these common standard formats, represent them in memory in a spatial data object, which is this combination of spatial elements, and also the user can then save and load the data representation to ZAR. ZAR is a standard format that is performant and cloud ready, and we save following the OM and GFF specification, which is becoming the new standard in microscopy and spatial omics. Finally, when reading the data, we represent the data in a lazy way. This means that even if there are data sets that are very big on disk, we can still represent them in memory and operate on them even in a standard computational environment. As I mentioned, the tables can annotate any number of geometries, and we can represent them on disk. And finally, on disk, we also represent different optional coordinate systems, which can portray the physical space, which uses micrometers as a unit, or the pixel space, which uses single pixels as units. And together with the coordinate systems, we represent also a variety of coordinate transformations that can be affine transformation, but also we are developing now ways to represent nonlinear transformations. This means that the data is not just saved in full, but also one can save the data together with coordinate transformations that will be required to proceed with the next steps of analysis. The in-memory representation is also compatible with standard Python libraries, and the user can easily implement downstream analysis starting from this. But also we provide some standard processing operations, such as spatial queries that allows to subset the various layers in arbitrary locations, feature aggregation, which allows the combination of signals from one layer to the other, and tiling operations that require steps to subdivide the dataset in smaller parts to then proceed with statistical learning approaches. Now, in the last part of the presentation, I want to show an example on the breast cancer dataset from 10x that I mentioned before. If you download the data from 10x, the data appears like this. We have a Visium, it's an eye field of view, and a Xenium immunofluorescence image that are not aligned. The first step is to make the data align, and we can do this with a landmark-based annotation procedure that we perform with Napari SpatialData, which is a plugin that we developed for the interactive viewer Napari. And now you can proceed with aligning, transferring the alignment also to the expression part. But also with Napari, you can not just limit yourself to landmark-based annotation, but you can perform freehand annotation. And here is an example where we share the screen to the iPad, in which we visualize together the Xenium and Visium dataset already aligned, and the image from the dataset, and then draw some precise anatomical regions. We see now some previously drawn anatomical regions that then will be used for downstream analysis. Finally, I want now to thank all the collaborators that helped in developing these packages and helped in writing a preprint that we recently submitted, in which you can learn more about what I just introduced in this talk. Thank you for your attention.